I'm Mary Parker, and welcome to this episode of Eureka's Sounds of Science. My two guests today are coming at the same issue from very different angles. Laura Gee, a colleague here at Charles River, lives with type 1 diabetes. She's coming at this chronic condition from a patient angle, daily monitoring and control. Jeffrey Millman, associate professor of medicine and biomedical engineering at Washington University School of Medicine in St. Louis, is coming from a research angle. The Millman Lab is striving to use cellular replacement therapy to potentially cure type 1 diabetes. They are here to bring these two perspectives together, and I'm glad they're both joining me today. Welcome, Jeffrey and Laura.
Thanks for
Great to be
having
here.
me.
Thank you.
Yeah, only two flubs in my intro. I'm very proud of myself today.
Yay.
So let's start with you, Laura. Can you tell us about your daily routine living with diabetes?
Sure, Mary. My daily routine living with type 1 diabetes is anything but easy, but it is part of my life. And it has been since I was just 7 years old. I wake up in the morning, I think about my day ahead, and I start planning. From the minute I wake up, I think about what am I going to eat for breakfast? What time will I work out? And all of these things sound like very small, but when you live with diabetes, it is something that really affects everything I do. For
Mhm.
example, when I come to thinking about what I'm going to have for breakfast, I have to think about how much insulin I need, what my current blood sugar is,
Mhm.
and really just, what I'm going to be doing after I have breakfast. There are many different aspects of a daily routine living with diabetes that must be considered no matter what I'm doing.
Mhm. I can see how the stress of that might add up over time. Like, even The Rock has a cheat day, and you can't really do that living with type 1 diabetes, right?
Well, uh, there are days, for example, okay, let's take a great example. We have Christmas coming, and
Mhm.
there is no way that I'm going to go through Christmas without having some homemade goodness, right? so, so of course, to me, that's a little bit more of a cheat because it's not something I would normally do. I usually don't eat a lot of sweets, but around
Mhm.
the holidays, they exist. So for me, having a cheat day just means having an extra big bolus of insulin
Mhm.
to help control my blood sugars.
Yeah, it still takes planning. It's not just something that can be overtly spontaneous.
That's right. Yep. It
Yeah.
all comes down to planning.
So, Jeffrey, what is your experience with speaking with patients? Are there any questions that you have for Laura?
Yeah, one of the things that I didn't really realize about what it's like to live with diabetes is the day to day and having to think about it. And this concept of you can't take a break from type 1 diabetes. You might be able to plan, like with what was just discussed here, but you can't just completely forget about it on any given day. And
Hmm.
so I guess I'd be really interested in hearing from Laura what it would feel like, what it would mean for you if there was a true cure or a functional cure for diabetes where you wouldn't need to think about the disease on a day-to-day basis anymore.
Honestly, it would be a dream come true. And it's such a big dream and something that I can't even imagine it. Like, I can't imagine not having a day where I don't have to think about, all those little things that I do right now. So to me, it would be a feeling I would not even be able to describe. I don't know if that makes sense, but that's how it would feel.
Yeah, I think we can probably, Jeff and I can imagine it, but we can't really appreciate what that feeling would be like.
Right. Yeah.
Yeah.
Yeah, and I think that's maybe one of the key takeaways here for members of the audience who uh, don't have to live with this disease is from my experience speaking with patients. It takes over your life. At least not takes over your life, but at least it's a major component of your day to day life. You can't stop thinking about it. And also, especially if you have if you're a parent with a child with type 1 diabetes, it's that kind of angst and fear can be amplified much more because it's your own child that is living with it, which is really what motivates the work that I do and other scientists in the field to be working in this space because we all realize how impactful it would be for patients if we were to come up with an actual functional cure for diabetes.
Yeah. And just so that people, in case they don't know the difference, the type 1 diabetes is the kind that you were born with. as opposed to type 2 or gestational, which might be more environmental?
It could be a little bit maybe more nuanced than that. So type 1, some people aren't necessarily born with it, but a lot of people develop it in childhood. But that
Hmm.
isn't true for everybody. There is a significant portion of people with type 1 diabetes that actually don't have it in adulthood. But the underlying cause of type 1 diabetes is a autoimmune attack where the immune system has failed to recognize the insulin secreting cells that are found in the pancreas as self and mistakenly attack and destroy those cells, meaning that the body is no longer able to produce insulin.
Hmm.
This is in contrast with type 2 diabetes, which is, I mean, there's a lot of nuances here, but typically is associated with obesity and insulin resistance, where essentially the beta cells end up having to work too hard in order to meet the metabolic needs of the patient and just can't keep up with how much insulin the patient needs. But so in the case of type 2 diabetes, oftentimes this can be managed with things like diet or taking drugs to increase the body's sensitivity to insulin.
Hmm.
For type 1 diabetes, that doesn't work because there just isn't any significant amounts of insulin being produced here. No amount of dieting is going to manage diabetes. And the only real way of managing type 1 diabetes is insulin injections.
Hmm. And you have to monitor your blood sugar to make sure that you're not taking too much insulin as well as not having enough.
Correct. It's very much difficulty here. It's the balance. It's not just maintaining a high level of a drug so it's effective, which is maybe how you would think about treating other diseases. It is a balancing act here where your blood sugar being too high or too low is both very dangerous to you.
So, before we get into your research, Jeff, Laura, I understand from browsing your online presence that you like to share about life with diabetes and healthy recipes and fitness tips, things like that. Why is it important for you to get that information out there?
I do really love to share about living with diabetes and recipes and things like that because it's so reassuring for me as a person to be able to see how other people that live with diabetes are living. For example, as a diabetic, quite often we feel insecure. We
Hmm.
are worried if we're doing the right thing. We're wondering, just how we fit in and are we okay? Little things like that because we just really want to do a good job. Most of us do anyway. And by sharing little glimpses of my life and maybe some recipes I've tried that I've enjoyed that had a nice low glycemic index that didn't skyrocket my blood sugars, for example, I feel like I'm contributing a little bit and also allowing people that might be a little bit more quiet or unsure to be able to see into my life a little bit and just understand and know that they're okay too. We're all in it together.
That's wonderful.
Mhm.
So, Jeffrey, let's get into it. How did your research come about? Where did it start?
Yeah, so I actually got into this research through a little bit of a different path than many people who are motivated to do research in diabetes. I was actually doing my PhD in, out of all things, chemical engineering. So I wasn't really focused on biology, let alone regenerative medicine for diabetes. And towards the end of my PhD, I was fortunate to have the lab I was in get a research grant from JDRF, which is the largest private funder of type 1 diabetes research in the world. And through that, and the patients and the scientists who are working in the space that I got to meet through it, it really showed me kind of a whole new area of research that I wasn't really aware of, being from a engineering background. And by speaking with them and learning about the field, I started to see how my kind of different perspective of science could approach the problems that were facing the field in kind of a new and creative way. And so once I got done with my PhD, I decided to completely change the trajectory of my career and training to be dedicated to coming up with a functional cure for diabetes. And since then, I have been working in this space now for over a decade, and have been really focused on the use of stem cells for the study and treatment of diabetes, particularly as a source of replacement cells that could potentially be transplanted into patients to mean that they wouldn't have to inject themselves with insulin anymore.
That's really funny that you come from a chemical engineering background because Laura, essentially, you're running chemical tests on yourself every day.
Every day, it's every day is another experiment, really.
Yeah,
And
absolutely. You're a chemist as well.
you can eat like the same thing 5 days in a row. You can do the same activities. You can have every single day exactly the same on paper.
Hmm.
But the way the body reacts every single day, it could be a little bit different. So it
Yeah.
is every day is another experiment.
And that's definitely how the cells can behave as well. And I think this is where the kind of chemical engineering mentality really intersects and synergizes well with the stem cell biology that goes on here. Essentially, the way that I have been approaching the problem of how to make these instant-secreting cells at the end of our manufacturing process is I treat it like a chemical reaction. Like in chemical engineering, the way we usually talk about things is that you have starting chemicals. It's a chemical A. You may go and create one, two, three, four, maybe more types of chemical compounds before you finally get to the kind of final chemical that you're wanting to derive here. And think about each of these as like separate stages. So you have
Hmm.
stage one, you go from chemical A to chemical B. Stage two, go from chemical B to chemical C. And you do them in a kind of serialized fashion to have the final chemical that you want there. So basically, what I've been trying to do for the last 10 years is apply that sort of mentality, but instead of having it just be chemicals, having the chemicals be in the final product to be cells. That we have stem cells that if we stick them into somebody's body, they're not really going to do anything, and figure out what are the correct intermediate cell types we have to make before we actually get to the final product here, which is pancreatic and secreting beta cell.
Okay, wow. So, how does the treatment actually work? The cellular replacement therapy, which is a mouthful, by the way.
Yeah, there's a lot of other terminology that we could use, but that might be the
That's
easiest one
fine.
to do.
I can deal
Uh,
with it.
in terms of the problem of controlling blood sugar levels in patients with type 1 diabetes, ultimately, this is an issue of there just not being enough healthy and secreting beta cells in the patient's body anymore. I mentioned
Hmm.
earlier that these cells are mostly lost due to a autoimmune attack. And so the basic procedure here is can we manufacture these cells artificially in a lab at a large enough scale so that we would have enough replacement cells that could be transplanted back into the patient so that these exogenous artificial manufactured cells can basically replace the function that has been lost in the patient's endogenous beta cells. And we refer to this collectively as cell replacement therapy. So the very first step of the process is what we refer to as germ layer specification. That's basically what kind of broad category of cell is the stem cell going to be called? The type of cell that we want to make. In this instance, to be able to make the secreting beta cells, they're called endodermal cells, definitive endoderm in particular. And so we have particular cues, mostly nodal signaling and Wnt signaling for people who care about those things, that instruct the stem cells to go from a cell that can become any cell type found in the body to only becoming cell types that come from definitive endoderm.
Hmm.
And so I mentioned that the pancreas comes from there, but also the intestines, the liver, the lungs come from that germ layer as well. But once the cells decide to become a endoderm cell, they can no longer become brain or nerve or skin or heart or blood or muscle or any of the other cell types that come from the other germ layers. So basically, we went from any cell type being possible to now basically only one third of the cell types in the body being possible. And we basically repeat that exercise through 6 distinct stages where we go from any cell type being possible to be produced from the stem cells all the way to just having beta cells and a few other pancreatic cell types as well being present at the end of the manufacturing process. So again, this is all done with soluble cues like proteins and chemicals there. It can be done at a fairly large scale where we're able to manufacture a few billion of these cells at a time. And really, the hope here is that with these manufactured cells, we would take probably about maybe 500,000,000 or 1 billion of these cells and to transplant them into patients. And as long as those cells survive and have access to the blood, they should be able to perform their function of sensing sugar levels in the blood and secreting a sufficient amount of insulin in order to keep the blood sugar levels at a normal set point, even after a meal has been consumed by the recipient.
And the cells can come from donors, right? They don't necessarily have to be the patient's own cells.
Right. So we look at both options here that we have done work where we have taken either blood cells or skin cells from patients with type 1 diabetes or some other forms of diabetes we study as well in the lab. And then we can reprogram these cells into the stem cells that we need to start off the process. And so these are a special type of stem cell that are called iPS cells, or induced pluripotent stem cells. So these cells, again, have the ability to become any cell type found in the body. And then we can give them our 6-stage recipe for converting them over into the insulin-secreting beta cells. Alternatively, you can use cells that are derived from a donor, or make beta cells and transplant those into patients as well. And that has the advantage from a kind of a manufacturing perspective. It's a lot easier and cheaper to take one donor cell line and make all the replacement cells that you'd want to generate and use from it for the hundreds, thousands, millions of patients that you'd want to transplant that into, as opposed to having to derive individual iPS lines from each potential patient, make the beta cells from them, and put them back into there. But we're pretty open to both concepts as being a way to go forward with this.
So I understand you have successfully done this in mice. I saw some news headlines recently about your lab. How has that gone so far?
Yeah, so we've had for many years now. We've published our initial protocol for making these cells, and a lot of various refinements to the process that have all worked very well in mouse models, and kind of the current generation, like the current 2021 versions of the protocol, as you can imagine, work a lot better than the prior reports that we had published in terms of being able to rapidly reverse diabetes in the mice, and being able to maintain control of blood sugar in these mice for essentially the entire remaining lifespan of the mice. But one thing as well that I don't know, I don't think we had a chance to talk about this when we were chatting before the recording started, but the original technology that I helped to develop for this has actually gone into patients now. And so this is back when I was a postdoc at Harvard University, starting off working with this. And I was one of the first authors and inventors of the publication that came out of Harvard in 2014, kind of showing the first significant report of being able to make these cells at all. And again, showed it to work well in mice. Since then, that technology has been licensed, and Vertex Pharmaceuticals is conducting a phase 1 clinical trial with these cells. And they actually released their results for the first recipient of these cells maybe about a month ago, showing that the first recipient who received a half dose of these cells is almost completely off of insulin. His insulin requirement has been reduced by 91%, and he overall is doing quite well and has avoided any severe negative complications with this yet. And it's also, maybe most importantly for him, because he had a lot of problems with this before the transplant, avoided complications due to hypoglycemia.
That's amazing. So, I mean, my next question was, what are some of the next steps to bring this research to the next level? But now it's kind of in the hands of the of the pharmaceutical company, and also it just has to kind of go through the regular scheduled drug approval process at this point.
For that version of the technology, that's definitely correct. But there's definitely a lot more work that needs to be done in order for this to be a true functional cure that can be deployed widespread in order to help over 1 million people living with type 1 diabetes in the US and beyond, of course.
Yeah.
So the biggest one that I am thinking about, that a lot of other people in the field are as well, is the issue of the immune system. And so what the Vertex clinical trials are doing in order to prevent their transplants from being rejected is that they are giving the patients immunosuppressant drugs, which are something that nobody ever wants to take. They come with their own risk of complications that, for many people who are living with type 1 diabetes probably would not be worth getting off of insulin therapy for. There are a certain category of people with type 1 diabetes for which that can make a lot of sense for. And like I said, this first recipient had a lot of problems controlling his diabetes and had been hospitalized quite a bit due to complications before receiving the cells in the Vertex clinical trial. But I and everybody else who are working in this space really want this to be a much more widespread therapy available to basically anybody. And
Mm-hmm.
the big elephant in the room that we have to address with this is the immune system. So how can we deliver these cells into recipients in a way that the cells will not be destroyed by the immune system, but the patient also doesn't need to take immunosuppressant drugs.
Mm-hmm.
And that's proven to be a very hard challenge to overcome. A lot of groups have focused on trying to coat the manufactured and secreting cells with various types of materials that will basically physically shield them from the immune system. And there has been some success in that, and I've actually published on that myself, but it seems to have problems scaling to work effectively in a full-grown adult with diabetes. In other words, it works well in mice, but doesn't seem to really scale to humans. And so,
Gotcha.
kind of the next thing that people are looking at, and that I personally have a lot of enthusiasm for, is this idea of using CRISPR gene editing to genetically modify the cells to be immune evasive. So basically, for the immune system to be able to tolerate the cells and not reject the cells, either due to the autoimmunity or due to them being from a different donor.
This is pure speculation from a non-scientist, but COVID has proven really good at evading the immune system. Could you maybe use its defense mechanisms as part of the process?
Well, it's funny that you say that. I don't know about COVID itself, but the approach that people are taking in the field is to look at other systems that are out there and seeing how they normally avoid the immune system, and then trying to just basically hijack that in a cell type that is not usually applicable for. And so I guess the two main kind of examples I can give for that would be how do cancer cells evade the immune system?
Mhm.
And there's certain surface proteins that are expressed on cancer cells like PD-L1 that help it to avoid destruction by the immune system. Or another example that I've heard people are employing right now is how are fetuses in utero tolerated and not rejected by the
Mm.
mother's immune system.
Mhm.
There's several layers of protection there, but there's certain HLAs that are expressed by the fetal material in order to avoid being rejected by the immune system. So, in other words, the COVID idea is not too far fetched in terms of how scientists are trying to approach this problem. And we've seen success with this kind of concept of trying to take known biological systems and understanding how it works in nature, and then applying it to translating it into actual biomedical needs. And this is
Mhm.
where CRISPR came from, for example. CRISPR-Cas9 is a defense mechanism of cells, but it so happens to be really good at editing DNA. And now it's a extremely common tool used in biomedical research and is being looked at for therapy as well.
So, Laura, do you keep on top of diabetes research? What do you think of the lab's work?
I do try, and I say try because there's always something new, and it's so exciting. What Jeff is talking about sounds super cool, and wow, I look forward to the day.
Yeah, imagine just like an off the shelf product. But, speaking of that, besides the immune system, I imagine that the other problem with, that type of future is scalability and literally manufacturing plants that can make these cells at a scale that would be needed by patients.
Yeah, and that's another really big challenge as well. So we have achieved a decent degree of scale, but I think new advancements need to be made more from a biotechnology angle in order to really scale it to produce cells for even any significant fraction of the number of people with type 1 diabetes. But even like 5 years ago, there wasn't really much like of a need to focus on scalability manufacturing because the first reports of the cells had come out and there hadn't been enough time for people to try to reproduce those results. Now we know that it's real. The technology is here right now, and now biotech companies are starting to take it seriously and are starting to think about these very practical questions, such as how are we going to make 1 million doses of this therapy?
Right. Yeah. I mean, this is a running theme in some of the other episodes is that the science always precedes the infrastructure. Just because Nikola Tesla or Edison or whoever invented a way to power things with electricity doesn't mean that the next day there were power lines sprung up all over the world. One has to kind of come before the other, and infrastructure always comes after.
I think it's a really good analogy, and again, I think it really just shows that how exciting of a space this is, and how fast things have progressed over the last few years.
So before we wrap it up, do you guys have any questions for each other?
I'm just thinking, Jeff, let's say you did cure diabetes. How would that make you feel? And what do you see the cure doing for, I guess, the nation? Big question.
But that might be a little bit above my pay grade, that second one,
Yeah.
but I'll do the best that I can. I mean, I fully expect to know how I will feel because I fully expect to see it in my lifetime, just because of how fast this has evolved and how well it is working. So maybe I give you my true response to it once it actually happens. But I guess I would speculate, of course, that it would be amazingly fulfilling. I mean, I changed my entire career trajectory to dedicate myself to this problem because I saw how important it would be for many people.
Thank you both so much for joining me for this. This has been a really, really thrilling conversation for me. And I really appreciate your time.
Thank you, Mary.
Yeah, thank you. I really appreciate it.